opportunity to talk to you all about open science and how it has to do with biopharma, biomolecular AI. My name is Kyle Tretina. I'm currently on the digital biology team at NVidia. As Tamkar said, I have quite a long history as an academic and doing multi-omics infectious disease type work before coming to NVIDIA. And I have just like a real passion for the use of AI, especially to study the very basic elements of life and matter. So you can find me on LinkedIn, on X, on pretty much every platform. I'm very easy to find, and I always love reaching out to new people who have similar overlapping passions. So would really look forward to connecting with some of you today. Specifically today, I think the context was set really well for this talk. I'd like to start with kind of a 10,000-foot view of what is happening when it comes to AI and biopharma, specifically in the context of drug discovery. I'm going to start with kind of a 10,000-foot view and kind of work my way in through 3 specific examples of research projects that teams within the overall digital biology research organization at NVIDIA have done. These are published examples, and hopefully I can sort of dive a little bit into the details there, but then also bring it up and think big picture about what it means, and then wrap it up and see where we can go going forward. Now, if you have my job, this is both inspiring and a big problem on the slide right now. So if you look at the number of AI models within biomolecular sciences over the last few years, it has absolutely exploded. Here, this is taken from a public meta-analysis that was done, but it aligns really well with review work that I've done looking in databases on both Hugging Face and GitHub. Number of published models for drug discovery, it's essentially grown to well over now 200 distinct models per quarter. So if you're trying to keep up with this space, it's not easy, and you very likely need some level of automation and AI in order to keep up, as I do. These are highly specialized tools for complex biological data. So some of these might be used to predict protein structures, to analyze biological imaging results, or single-cell transcriptomics, and you know, the fact that there is both very many of these models, as well as the technical depth and exploration happening from a mathematical perspective within each of these, and how they're inspired by other domains, it's really awe-inspiring. We're seeing a total shift in how, especially deep learning, is being applied to biopharma just in these few years. Simultaneously, along with that, adoption has been very pervasive. 38% of industry researchers report substantial or extensive use of AI in this research done by BCG this year. And so if you think in a historical perspective, it was really 2025 where real, like, in-depth, meaningful adoption of AI started in biopharma. So I think this agrees with other reports that I've seen from other research being done in the market, as well as my own personal experience, right? I get to talk to organizations throughout the industry, from startups to software vendors to research organizations and academics on a pretty regular basis. And at a really high level, this creates a really powerful virtuous cycle. So we have this explosion of new tools that enables researchers to tackle previously intractable biological questions, or to accelerate or save time when doing old problems. And their widespread adoption can now generate the feedback and the validation, and in some cases, even the new data that are needed to build even better models. So for biopharma, this means that AI is rapidly moving from this very peripheral R&D support function into really a core competitive necessity in a lot of cases. And this was the year when we really saw that big shift happen. Now, the ability to build, fine-tune, and deploy these specialized models against proprietary data is increasingly, in my own conversations with organizations, becoming more and more of a primary driver of R&D success than it ever has before. And so I'd like to go into a little bit more detail about, based on my own personal experience, why would people even care about AI being open source within this really highly specialized domain? And so I'd like to, I call this a rocket diagram. It's supposed to look like the underside of a rocket. So, okay. So the transition of biology from an observational science into an engineering discipline, which is what we really kind of hope it will be one day. Well, that's why we call ourselves digital biology over on this team. It hinges entirely on the adoption of open source artificial intelligence, and that's a core belief that we have. For the pharmaceutical industry, the most critical asset, as I hear some of you talking about in the chat, is proprietary data. A company's chemical libraries and patient genomic data, for example, these are really the crown jewels of those organizations. And in some cases, relying on a black box AI model through an API presents an unacceptable risk, depending on the use case. Open source models really pretty quickly solve, depending on the details of and level of openness, the data privacy and security challenges that a lot of these organizations have, by allowing them to bring the intelligence to their data rather than sending their data to the intelligence. So by downloading models to run within their own secure perimeters, pharma companies can ensure that sensitive IP never leaves their control. I know in my own experience as an alliance manager previously at AI-driven drug discovery company, in interacting with pharma companies, having their data run through another company's servers was pretty much a non-starter for most of our projects, especially for early proof of concept type experiments and work. And so this is kind of the starting point. I would call the table stakes, and why OpenAI is pretty immediately appealing to organizations in biopharma. Now, this control extends to the licensing customization of the technology, as I alluded to there. So in the high stakes world of drug discovery, companies often require like the freedom to operate without the fear of vendor lock-in or licensing disputes that could jeopardize a drug program years down the line. So furthermore, biology so far has proven to be far too complex for a single generalist model. A lot of the models within biology that are being used, especially for biomolecular AI, have pretty narrow functions, although that's rapidly changing. Open source enables what I call an AI foundry approach, where companies can take a foundation model, like an ESM2, and fine-tune it on their specific wet lab data. And this transforms generic capabilities into a specialized model that can understand the company's unique therapeutic targets or subset of molecules, and ensure that their model is not just intelligent, but actually relevant to the specific biological programs being solved. And from a scientific standpoint, open source can also help to provide some of the rigor required for certain regulatory environments. Transparency, in some cases, for example, as you're preparing an IND report, it can be, well, it is in that case non-negotiable. Really, when human safety is at risk, transparency is often required. Researchers and regulators must be able to inspect the code to understand like how the prediction was made in some cases, rather than just trusting the algorithm. And this connects directly to reproducibility. So having a closed API might change without warning. rendering previous results unrelated to new results. And open source guarantees an experiment run today can be replicated exactly within 5 years from now by using the same version. And that stability is often essential for the long timelines of clinical trials. And finally, open source creates a unified ecosystem that accelerates the speed of discovery while lowering the cost. And this, to me, is easily the biggest reason why I think we're involved with open source AI in this domain. So by establishing common standards and providing new open tools that allows different software from molecular docking to protein folding, to interoperate seamlessly, and this democratizes the state-of-the-art technology. That allows the whole global scientific community to actually contribute to the innovative research that lifts the entire industry. And by moving expensive wet lab screening increasingly into digital simulation, we can explore the vast universe of drug-like molecules for the increasing the cost of electricity, ultimately delivering life-saving cures faster and more efficiently than ever before, hopefully. And so it was mentioned, I do have a blog. I often write about projects in collaboration with some of the researchers that do those projects. Several of them are listed on the page today. These are actually, this is the NVidia technical blog resource within our developer domain. All of these are open source, available on GitHub. Where there are AI models, we always release, we make them as open as possible, including, when possible, all of the data that was used to make the model, and in many cases also tools that people in drug discovery rely on. So this is all pretty easy to find, and I'll have a link at the very end of the slide for you to point to be able to access them yourselves and test them out if you'd like. I like to group them into 3 categories here, although there's quite a few different ways you could group them. I like to think of generative design models like Tretina, which is I'll be talking about today, for sequence structure co-design with proteins, as well as GenMol for generative molecular design. And the idea here is that we're releasing, hopefully, something that can actually contribute to the comprehensive ecosystem of open models, tools, and recipes that the ecosystem's building. And we want to actually focus on highly computationally difficult problems from small molecules, DNA, RNA, and protein structures as well. In addition to that, we also have predictive evaluation models. So within these different classes of models, especially depending on the architectures, there's not always a generative process that happens within those. And also based on the use case, you may not want to just design a molecule. You may also want to predict its property, which different properties of those molecules, which is increasingly becoming more and more important as people rely on these types of models more often. And then there are toolkits and libraries that people rely on every single day that we're often moving into similar types of technology stacks as these other models, as I'll talk about later on. So essentially what I'd like to do here is use these one project from each of these 3 categories as kind of examples where we can kind of dive into the nitty-gritty of how they work and hopefully show how open source AI is actually changing drug discovery with really concrete examples. So first, I'd like to start with the generative design models, talking about the problem of generative biomolecular design. This was introduced earlier on, but the central challenge that we face with generative biomolecular design extends far beyond simply generating novel sequences. Although, that's kind of the fun part to dream about, in my opinion. As the visual on the left illustrates, so moving from something like a raw genetic code to these complex 3D structures, our true goal is to engineer molecules that are functional, stable, synthesizable, and have other qualities like a drug. So we're attempting to navigate this really difficult path from something that seems simple on its face and kind of wandering out into this vast, complex universe of viable biological molecules and machines. And this path is blocked by really fundamental hurdles. So first, we're contending with this really astronomical search space. The combinatorial magnitude here is functionally unsearchable. So even a simple 100 residue protein represents, something like 20 to the 100 potential sequences. To make matters worse, we're navigating in this infinite space using sparse, noisy, and often biased data. Even if it is internally generated, our current experimental knowledge represents really only a tiny and clearly non-representative fraction of the biological landscape, which can make training reliable models exceptionally difficult. And even when we identify something like a candidate, we can call a hit, it must survive the gauntlet of physical and functional constraints. So a sequence must first obey the rigid laws of physics to fold correctly, for example, before it can even attempt to satisfy complex functional requirements within a biological context. This is further complicated by multi-scale coupling, where the properties are often interlinked across levels of biology, from all the way down from quantum interactions up to the atomic level to the macroscopic biological functions, and then organs and organisms. It's pretty complex. So finally, we all often also hit the bottleneck of computational cost of feedback. So validating a design is an absolutely critical step that evolves over the life cycle of research, early R&D, and high fidelity simulations and wet lab experiments are slow often and expensive, and it's difficult sometimes to support rapid iteration. So in short, we're trying to find a few specific needles in a combinatorial haystack and navigating using a biased map while strictly obeying the laws of physics, and all while every single test of our progress comes at often a high cost. And so specifically, I'd like to dive into Tretina, which is a project that was recently launched by our generic group here. And looking at the specific problem that it solves. So the next frontier in protein design is very increasingly moving from the generation of not just coarse-grained backbones, but actually the entire molecule at the atomistic level. So what we're showing here on the left is a string of amino acids, which then fold in various ways to form the backbone structure of a protein. And in order to generate fully atomistic designs, this requires modeling the joint distribution over both the sequence on the left and the all-atom 3D coordinates on the right. Now, this is a very difficult task for a couple of reasons. So first, there are mixed modalities here, so it involves simultaneously generating discrete data, like the sequence on the left, and continuous data, which are essentially, you can think of like the coordinates on the right of the backbone structure. So there's also what's called variable dimensionality. The number of side chain atoms is sequence dependent, which means that the total number of atoms actually changes during generation, which is a really severe challenge for standard deep learning frameworks. However, solving this is really important. So true protein functions not defined just by the backbone alone. It's mediated at the atomistic level by these side chain interactions, and therefore modeling this joint distribution is essential for enabling high precision functional designs, like designing a novel binder or an enzyme, which depends on the exact geometry of the active sites. Yeah, it's hard. So Lepretina's core innovation here is a partially latent representation. So it's designed to solve the fundamental challenge of all atom generation, as I just laid it out. So it essentially couples the generative problem into two components. So first, there's the backbone, explicit backbone. So the core structure is represented explicitly by the alpha carbon coordinates, and then the amino acid and all the corresponding side chain coordinates are compressed into what are called per residue fixed size continuous latent variables. Don't worry, I'm going to bring this up in a little bit. This representation is learned using what's called a variational autoencoder, certain architecture of model. And the encoder learns to capture all the sequence and the side chain information from a fully atomistic protein, and then the decoder then deconstructs the full atom protein condition on both the explicit backbone coordinates and the learned latent variables. So this design is really critical. It converts the complex mixed modality and the variable size problem into something simpler, fully continuous, fixed dimensional, and making it highly amenable to generative modeling. Okay, so the generative component of La Tretina is a partially latent flow matching model. As mentioned, this model is trained to approximate the joint distribution over the explicit alpha carbon coordinates and the fixed size continuous latent variables. So I'm happy to talk offline about the details of this model and connect you actually with the research team, but I'd like to actually focus on what this means at kind of a higher level. A key breakthrough of this framework is its unprecedented scalability, which is enabled through that partially latent representation that I kind of breezed through there. This design avoids the computational memory bottlenecks of full explicit atom modeling by compressing all sequence and side chain details into a fixed size per residue latent variable. So, what does that mean? This architecture basically allows La Tretina to generate diverse, co-designable, and structurally valid atomistic protein at length up to 800 residues. For context here, here's a comparison of what's called co-designability. So looking at designing both sequence and structure at the same time over various residue lengths, so the length of the protein. And so you can see the green line is above all of the other lines. So whereas other models often crash around like 400 amino acids, La Tretina is still making quite a lot of co-designable molecules all the way up to 800 amino acids. And for context here, it's very, very, very common for people to want to design molecules larger than 400 amino acids. So many molecules within the human proteome are hundreds or even thousands of amino acids. And then if you think about complexes, you very often want to generate complexes or are interested in complexes that are many thousands or even sometimes tens of thousands of amino acids in total length. So beyond co-designability, we also wanted to assess the fundamental biophysical validity of the generated structures. There's this metric called mole probity. La Tretina generates structures that are significantly more physically realistic than all the baselines and achieves a superior overall MP score. The bottom line here is like it's able to have very low physical clashing, which means more physically realistic models. At a really high level, the paradigm in protein modeling that this illustrates is that we're shifting from prediction, so think AlphaFold to kind of breaking through and generating experimentally relevant single monomer structures for really well-defined proteins, to something much more complex now. We're shifting to a new generation where we're thinking about all atom prediction, de novo generation, and what is something that represents proteins prediction that would actually be a little bit closer to what we would actually want in a drug. And this new frontier is driven by applying scaling laws to generative models. So The next critical step here in like what we want to do next is moving from these coarse backbones to co-designing the discrete sequence and continuous all-atom structure like we're doing here. And this progression from prediction to all-atom design is a foundational path towards that more generalist model in this space that I mentioned before, where hopefully one day we'll be able to prompt these models with a function to generate a novel, viable protein that can actually be realistically tested in a more complex environment. Okay, so now I'd like to move to predictive evaluation models. Okay, so if I lost you with a little bit of that before, now would be a good time to wake up again. If generative design is the engine of discovery, property prediction is the steering wheel. The companion challenge to creating new molecules is the ability to accurately evaluate them. Arguably, this is the most important application of AI in BioPharma, although that's probably someone who works on toxicity prediction that convinced me of that. Fundamentally, we're attempting to learn a mathematical function that maps any point within this vast chemical biological space directly to its functional outcome. However, biology presents a unique set of obstacles that makes this task really difficult. The first barrier is the disconnect between the size of the search space and the data we possess. I mentioned that before. We're operating this vast chemical biological landscape where combinatorial possibilities are astronomical, but just as importantly, the labels that we rely on to train these systems are often weak ground truths that fail to capture the full picture. So for example, we often optimize for metrics like an IC50 value, but this is like merely a single low-dimensional scalar trying to summarize a complex dynamic biological event. And we're asking our models to learn high fidelity reality from these like low fidelity proxies. Finally here, the landscape is actually, I would say it's treacherous. We're dealing with high dimensional nonlinear targets where simple linear regression can often fail. And the entire system is governed by pretty complex physics. So the next model that I want to talk about here is part of a group of themes that can be somewhat controversial because the vision for this group of models is huge. And the things that it can do are truly amazing, but where we want to go with it is far beyond where we are. This is code on FM. And the vision here that I was talking about is the virtual cell. And I know sometimes talking to scientists or even just saying that word can make some people upset. So I want to be a little bit more concrete around what we're talking about at the moment. I really like this paper that came out this year. It was I think late last year about the vision for the virtual cell, or one particular vision for virtual cell. And it really connects to, I think, the ambition of the phrase digital biology, where we're conceptualizing this not merely as like a database for recalling information about biology, but really as a learned simulator. That's the goal. A multi-scale, multimodal foundation model built directly from biological data that you can query to learn about biology. And as you can see in the architecture on the screen, we're mirroring the complexity of life by building a hierarchy of representations that allow us to predict behavior across different scales of biology. So La Tretina, we just talked about, begins at the molecular scale. And there we're using, very often it's transformer-based models, but in the case of La Tretina and others, that's not the case. So to encode, but what we're trying to do is encode primary sequences. So DNA, RNA, proteins into this continuous vector space and create a molecular, hopefully one day, a molecular universal representation. So moving up to the cellular scale, the idea is that hopefully one day we can integrate these molecular vectors with other high-dimensional modalities like omics data, cellular imaging. And this fusion creates a cellular universal representation. And I hope you can kind of generally see where we're going with this as we are slowly building our way up the scales of biology. So then to the multicellular scale where we treat cells as nodes within a graph structure, and using these techniques like graph neural networks, for example, maybe one day we can model the spatial organization and complex cell-cell interactions that define tissue function. However, in addition to that, a representation is really only useful if we can interact with it. So we'll need virtual instruments there to come into play, and think about things like how are we manipulating the system in a way that we can learn from it. But today, this is really just a vision. As it says on the screen, we're pretty far from this, but it's such a powerful, compelling vision, I think, at least in my opinion, that it's worth every single step in fighting for and working towards. In Codon-FM, in this particular case, it's a family of foundation models that we launched recently. And the idea here is to test some of the scaling, what we would find are scaling laws of biology by thinking specifically about codons. And so I see in the chat here, I don't think I have to explain what a codon is, but basically within an RNA molecule, specifically those that encode for proteins, there are triplets of nucleotides that encode for proteins within that RNA. And in this particular case, we trained one of the Codon-FM models called NCodon on this massive, diverse data set sourced from the NCBI RefSeq database. This corpus, it consisted of over 130 million coding sequences for more than 22,000 species, with this intentionally broad phylogenetic distribution encompassing bacteria, archaea, fungi, plants, and others. The architecture is transformer-based encoder that operates directly on codon tokens. So not individual nucleotides like many, let's say protein or other maybe DNA models are trained on. So the unit of logic there is a codon, not a nucleotide. The pre-training task is a masked codon prediction objective, which is analogous to masked language modeling, NLP. As you can see here, let's see, the model learns, hopefully should learn to predict the probability of a masked codon given the surrounding codon context. So it's still, you can think of it as like next codon prediction as the next token. And this hopefully we found will force the model to learn the complex context-dependent rules of codon usage bias and sequence grammar across the entire tree of life. And so we, here, we're evaluating the quality of the representation learned by encodon at different scales. This is called synonymous codon confusion. This metric is derived from the mask prediction task itself, which measures the frequency with which a codon mispredicts one synonymous codon for another. And so, as you might be able to see, a clear scaling law is evident, meaning colors get redder as you go up, meaning that the 80 million parameter model shows high confusion, meaning it often fails to distinguish between synonymous codon, and as the model capacity increases to 1 billion parameters, towards 1 billion parameters, the normalized confusion score progressively decreases. So less confusion is good. This demonstrates an emergent property. So the larger models are, we think, actually learning more of the nuanced context-dependent grammar of codon usage and capture some of that, hopefully, like regulatory specificity that dictates which synonymous codon is quote-unquote correct, not just the amino acid that it encodes. This is also seen in the embedding space shown below in these UMAP projections. I'm not going to emphasize that because I don't like UMAP projections personally.
There is a question in chat, Kyle.
Yeah, no problem.
Emma asks, can Codon-FM replace or augment the BLOSUM 62 metrics?
That is a really good question. I think, I'm trying to look at the slides here, that is addressed in the paper. The idea here, let me get to the end of this section. If I don't answer it, let me know. I think it's somewhat addressed. So you might use one of the BLOSSOM matrices if you're designing mRNA, and that is one of the tasks that we do evaluate on here. So you know what, I can actually, here we go. Okay, well, the next slide is one. Actually, great transition. Here we go. So to evaluate if encodon captures these functional constraints beyond synonymous changes, we benchmark it on missense mutation prediction. The evaluation is performed in what's called a zero-shot setting. So there's no learning loops. And as illustrated, we score each variant by computing the log likelihood difference between the original codon, reference codon, and the new mutated codon to give the full sequence context. This score basically quantifies how surprising the mutation is to the pre-trained model. And we applied this method to a large de novo variant cohort from this database called Deciphering Developmental Disorders, DDD. The bar chart shows the statistical separation between pathogenic and benign control variants. So this is a discriminatory test. And as you can see here, the encodon model achieves state-of-the-art zero-shot performance and outperforms the other listed unsupervised models, including protein-level models like ESM2 and other RNA-based baselines. And so the idea here is that by learning the contextual grammar of codon usage, it can implicitly learn these high-level representations that encode protein constraints and functional relevance. This is just one example. There are other examples in the synonymous variant prediction as well that we show here. This is a slightly different context where we're looking to distinguish pathogenic from benign silent mutations in the ClinVar database. This is a really hard benchmark. Any functional effect is likely not due to change in the protein sequence in this particular case, but to ensure a robust evaluation, we used a rigorous stratified subsampling approach that you can find in the paper, where basically we performed 50 iterations that match pathogenic and benign variants for codon type, gene position, gene level constraint, and local mutation rate. All right. That's far more detail than you probably needed to know there. What I did wanted to also point out here, so we've already talked about missense prediction, synonymous variant prediction, but we also looked at translation efficiency prediction as well. So whereas the previous two might have context in a diagnostic type setting, this might be super relevant for actual mRNA production and design. So you can think if you want to design an mRNA molecule that's going to be really highly expressed in a cell type of interest, or if you have an RNA therapeutic that you want to be able to be commercially manufactured at a really high expression level, this could be useful. And here, what we're looking at on the screen here is using a zero-shot embedding from each model as features for a random forest regressor. And basically, we found that the 1 billion parameter model achieves the highest explained variance with that R-square scored exceeding 0.5. This is, I think, kind of a big deal. People, if you go back and read the codon bird paper, this bottom model here, I think quite a few people that I talked to had sort of given up on scaling laws in this type of model, looking at codons, and even seen it as maybe a dead end, although that might not have been the case of the leading thinkers in the area. And so to actually find that by simply scaling the size of the model, the data, scaling the AI, you can actually get better performance here is pretty amazing.
Kyle, another question in chat. Bradley asks, you mentioned 3 types of predictions. There was the missense prediction, and the last one was translation efficiency prediction, and there was one more in the middle.
Yeah, that would be synonymous variant prediction.
Does that answer your question, Bradley? Perfect, thanks.
Awesome. Okay, so the future of codon models. Basically, what we're sort of building on here is this one insight that clearly synonymous does not mean silent. I think if you've done work in molecular biology, you've probably bumped into this before. But 4 years, it was assumed that if 2 different codon sequences encode the identical protein, that they're functionally equivalent. depending on the scale of biology that you're looking at. We now know that this is false. Different codon choices can lead to functionally distinct biological outcomes, such as drastically varied protein expression levels, which might be the most common mechanism there. And we're now using AI to decode this hidden layer of biological regulation. So this research with Codon FM directly tested this by applying scaling laws. So training models from 80 million up to 1 billion parameters, we demonstrate that the model performance scales predictably. And as we scaled up, we observed this ability in larger models to basically deconfuse these synonymous codons. And so what we're really hoping here is, as we build an understanding at these lower, sort of smaller molecular levels, that we can bridge the gap between the molecular and cellular scales. And that's going to require quite a bit of experimental validation, where we confirm experiments of different types, like perhaps ribosome profiling or synthetic mRNA design, and actually be able to use different types of multimodal data and integrate them to connect these molecular models into more of a dynamic cellular context. And this is where the rubber really meets the road. There doesn't seem to be many, if any, useful models in that space yet, but the quality of the models is improving pretty rapidly. And there is reason, I think quite a few technical reasons for excitement here, that that this is a line of work that's worth diving into more. And there's a tech blog on each of these that you can link to on the site that was shown earlier to read more. Okay, so the final section here that I'd like to bring up has to do with toolkits and libraries. I think this is really sometimes overlooked. Just looking at the impact of OpenAI on drug discovery and research in biopharma. And if I were to explain to a scientist why I thought this is an important area to talk about, I would just say, we can't really view AI as a standalone tool. If you ever hear like Jensen talk, like it requires full stack thinking. Achieving the speed and scale necessary for digital biology to explore and generate insights into biology, it really demands a more cohesive solution that spans the entire computational spectrum, from like the raw hardware all the way up to the high-level domain applications that are used very literally by scientists. And this integrated ecosystem is essential for unlocking novel breakthroughs and ensuring that software and hardware work together in perfect concert. And this foundational stack relies on highly optimized software libraries. So these libraries feed into the SDK layer, and ultimately this full stack abstracts away some of the computational complexity at the application layer, but it allows us to essentially co-design the upper layers by understanding the lower layers. And why is that important? I'm taking a screenshot of a slide that Jensen talked about here. I think it was a really important point because it does impact our everyday work as scientists. And that is that there's actually two revolutions going on. It's not just an AI revolution. There's also an accelerated computing revolution going on. And both of these are directly impacting scientific research, really at an everyday level if you use a computer. When I was more and more so like in my postdoc, this isn't something I think thought about a whole lot because I was so obsessed with the biology, but it is critical to understand basically that this is happening. So in the place of previous like general purpose computing, accelerated computing is providing pretty dramatic speed ups for computationally intensive workflows. And so I'm going to provide just one example of that within the cheminformatics space called NViMolkit. And so cheminformatics toolkits are essentially like the bedrock of molecular analysis. So they have historically, however, struggled to keep pace with this exponential data for certain types of workloads, and how that data is scaling now, which requires, in some cases, more modern deep learning pipelines. And RDKit is one of the most commonly used, I would say, the most commonly used toolkit in this space. I would actually argue its dominance is pretty undeniable, with millions of downloads on Conda Forge, and recent development activity within its ecosystem is like live and thriving. And so while RDKit's language everyone speaks, I think there are very specific uses of it for larger workloads that require it to essentially have performance that goes beyond what CPU-bound systems that scientists often have in their labs can process. And so we wanted to work with the amazing people, Greg and team that started this library, to see if we could create an accelerated version of it. And I'm just going to summarize it here. At kind of a high level, some of the early speedups that we're seeing. The primary goal of this effort was, as I mentioned, to accelerate RDKit functions that have specifically become computational bottlenecks for specific high-intensity pipelines. And so we've done that through both acceleration of the algorithms on the CPU, as well as porting what we can to GPU-accelerated parallelism. So you can see some of those functions here. These are like really core functions. I want to be just conscious of the time here and just kind of dive a little bit more into one as an example. And really at a high level, the architecture of NVidia Kit is engineered to deliver extreme performance without forcing to rewrite the code base. And so what this team is doing is at its core, we've essentially implemented a robust library using C++ and CUDA to handle the heavy lifting. And as illustrated in the diagram, it functions basically as a seamless drop-in replacement. So that's the goal, at least, to be as comparable to the API as possible. So the Python interface operates essentially as a pass-through that interfaces directly with the native RDKit C++ API. And if you look at the code comparison on the bottom right, you'll see the transition. Like, hopefully, the idea is that it should be trivial. We maintain function signatures and dependencies that are nearly identical to what people use today, allowing you to simply swap in the library and immediately unlock accelerated computing. Now, crucially, this hybrid open source structure is specifically optimized for batched processing by moving from processing single molecules on a CPU to massive batches on a GPU. Basically, it can provide, as I showed on the last slide, that in some cases, magnitude speed up for high throughput workloads. Now, this is being provided as a separate standalone library, more for operational purposes, but this was recently announced and presented at the most recent RDKit user group meeting, and so there's a lot more to say here. Okay. I want to be mindful of the time here. The shift toward scalable open foundation models is very clearly being widely adopted across the biomedical ecosystem. And I think everything that we're showing here represents different projects where we're applying this approach to accelerating what needs to be accelerated, or taking AI and sort of advancing it and making it as open as possible to lift the entire ecosystem for the reasons I described before. This adoption of AI represents like really this deep collaboration between these leading AI institutions across the entire spectrum of drug discovery and medical research, with very often like the people actually doing drug discovery. And so at a really high level, there's something kind of societal and profound about the changes that we're seeing today in the meshing of different cultures, even within science and the way of life and doing research today, from major pharmaceutical companies and cutting-edge biotechs to distinguished academic and clinical research centers. I do think that there is a somewhat unified push toward having open standards in many of these, but it's frankly difficult. With the pace of AI being released, the models that are considered state of the art are changing very rapidly in most cases by most benchmarks, and being able to have standards that and either competitions or benchmarks that people actually believe in is not easy. If you thought it wasn't easy for ChatGPT and for others, it's way harder here, where we can't even agree on which assays are best for our specific questions. And so in order to make it to kind of the next phase of this field, I think a collaborative environment is going to be really essential for refining the models that we have to ensure that they meet rigorous real-world standards that are required for these really complex therapeutic and clinical applications. And none of this research that I showed here or before would even be possible without the tireless dedication of quite a few digital biology teams at NVidia. So I want to apologize for not trying to cram them all onto one screen, and instead just showing the slides so that people can look up the projects and Google them. And if you have interest in a specific project, I'm happy to put you in touch with that team, or at least refer you to resources to get your questions answered about them. But as you can see here, AI is influencing all of early R&D throughout the whole research and discovery pipeline, through all of the traditional steps. And it's becoming a part of our life in a way that I didn't anticipate when I started this industry. And I think is incredibly exciting in some ways, but changing not only how we work, but also how we interact with other researchers in order to take a big advantage of it, will require some discomfort and social change in some places that I think we have to be open to. I know, when I was a postdoc, for example, I really got good at certain assays, and it felt really good to do them many times and continuously push forward the edge of knowledge. And it was hard to learn new things, and sometimes to change the way that I approached a problem in order to reach the next level in my career. But I hope we can all be open to whatever's next. And I think we have some ideas about what that next is, but I wanted to leave a few minutes here for discussion at the end. And that's it. You can find all the code and projects at the link at the bottom of the screen. Thank you.
Thank you very much, Kyle. This is a lot of information, and I really enjoyed the talk, partly because a lot of this is very foreign to me, but it was very interesting. We will officially end the meeting in case people want to leave, because we are at the end of time. So thank you again very much. However, there is a question by Emma, and I have several questions for you as well, Kyle. So if you're able to stay a little bit longer, for those who can stay and who are interested, we can continue. How does that sound?
Sounds good. I'm here. Let's do it.
Okay, awesome. So Emma, why don't we get your question first? So thank you again, everybody, for being here, and please feel free to stay or leave as you see fit. So Emma's question, do you want to read it out, Emma, or do you want me to?
Yeah, just when you're talking about speed ups and I work with Whittle workflows, and so we've been appreciating the Parabricks projects, and it sounds like what you were describing is a similar effort.
Yeah, absolutely. I work with the Parabricks team every day, so absolutely love their work. They are Yeah, really. I was actually a Parabricks user before when I was earlier in my career, before they were acquired by NVidia. But yeah, that is a team that just really continues to grind on extremely important problems in data analysis. And there's some evidence that biology is hitting a data wall right now. Well, one way past that is to make that really not just the generation of data, but also its analysis cheaper and faster. And so they're directly applying that to that problem, like with almost a religious fervor, especially for workloads that matter for the genomics community.
Nice. I just want to jump in to note that I myself am working on here at Fred Hutch, we're working on a Wild's Whittle library, we call it. So, Nextflow has this huge library, and I know Parabricks has Nextflow workflows as well. But
Yes.
anyway, we can work closely to add Parabricks based whittles to our library that then we're trying to share with other institutions that we collaborate with and with our scientists here. So it's very exciting that you've been working on this, and that's something we've been paying a lot of attention to. So just putting that out there.
Awesome. Yeah. Anyone here is doing really any kind of secondary analysis, please consider Parabricks. I think saturation is pretty good with them. They're more of one of the more mature products for accelerated computing and bioinformatics. But the ecosystem is really growing, and all the big sequencing companies and others are either using it or looking at it. So yeah, it's an exciting place to be. I did my PhD training at the Institute for Genome Sciences at the University of Maryland with half the group that split off from Tiger after the Human Genome Research Project, and so genomics is kind of where I was this is going to date me. I got into science because of the Human Genome Research Project, reading papers about it in high school, and then I finally got to work with some of these people in grad school, and it was the most inspiring thing and really hard to not just nerd out every day going to lab. So yeah. So genomics has a special place in my heart. I don't focus on it as much right now, but yeah, really good stuff. Nice to meet you.
Thank you, Emma, for your questions, and Kyle for answering them. So I have a few questions which are not as technical as Emma, because this is not exactly my field. But one thing I was wondering was, when you create these foundation models, do you use some sort of grounding techniques based on physics, like, for example, just the triangle inequality where atoms can live, right? So do you use techniques like that?
Yes. There's, and this is a very important area of research, really all across biomolecular AI. If you look at, for example, E3NN, so E3 neural networks, we're doing quite a bit of work there with our Cuckoo variance library to accelerate those. Just basically looking at symmetry in molecules to hopefully reduce the amount of compute required to design them or to make predictions about them. I think in the future there's a few different sort of high-level directions that this will continue to be important. It's already been really a driving feature of virtual screening at scale, where all the existing methods have been using physics basically for, I don't know, decades or 100 years, something like that, that we, as we know them today. And basically AI-based approaches, especially what are called MLIPs, basically are hopefully taking those methods and just making them faster. And the hope is that you can make them faster at an accuracy that is acceptable. And this is one of the most important kind of races within drug discovery today, is has to do with virtual screening. You have companies like, the Schrodinger's out there that are kind of leading with their amazing force fields that they're working on. And so yeah, there's AI going into physics to sort of take the entrenched physics methods and hopefully make them faster. But then there's also physics going into AI. where, you know, I was just talking about La Proteina, sort of modeling proteins at the atomistic level, and trying to integrate representations that actually represent the physics better, so that when you generate a molecule, it respects the laws of physics kind of right off the bat. And so you don't have to generate as many molecules and like throw out 90% of them like we had to do at the very beginning. But we actually have something that at the very least looks like the type of molecule on its surface, like just, you know, roughly, to give us a good starting point. So yeah, AI is going to physics, physics is going to AI. I think in the future, every single biological method will very well integrate some aspects of physics into it, especially as we are working our way up the molecular scales of biology. That'll be required. There's some people, though, that think that we should go top down. That's another view that's out there. I don't hold it, but yeah, that's just another thing. We'll see what happens.
Yeah, and then a related point is that ultimately all of these molecules, the atoms, they behave quantum mechanically, right? So a natural question then would be, would quantum computing have an advantage here? And where are we in that regard?
Yes, absolutely, 100%. Quantum computing is coming. We're investing as many biopharma organizations, you know, we don't do drug discovery. We're a, you know, AI platform company, but yeah, quantum computing is coming. When it comes to biopharma, there are some really interesting, cool toy use cases out there. I'm not aware of, although, you know, there are some companies that say they're, you know, using it in production for interesting chemistry problems. That is very small scale and on kind of dedicated, they're called like ASICs chips for specific well-defined areas. So it's not here yet. When it does come, it will, there's different ideas about how that's going to impact how research in biopharma is done, but I think the most compelling one to me is really just having to do with it being almost another accelerated computing, where, you know, it seems like the cost of and complexity of using them and running them will help us get to the next level of molecular dynamic simulation, for example. And so they'll have to be complemented for more coarse representations by, like, let's say GPUs or CPUs. Basically, I think it'll be a new class of chip that will complement what we already have. And then depending on the problem, you'll apply one and or the other, or maybe together in some combination in order to get a better answer. Sorry, does that make sense?
Yes, yes. Like I'm thinking that cyborg and centaur model actually, which is like currently they talk about it in human and AI, but like the same thing might apply to, you know, like AI, like the traditional computers and quantum computers as well.
Yeah, that's a really, I actually really like that analogy. At its very core, though, I think emotionally, quantum, like chemistry is quantum. And so it passes the sniff test. And that's kind of where we're at. And so everyone's investing in it, you know, in order to be there when it happens and to understand it and be able to use it when it really explodes. But yeah, we're just not there yet, especially for biopharma.
I have one final small question just because of a side comment you made. You said you don't like UMAP. Why is that?
Yeah, there's I think there's some disagreement around how interpretable UMAPs are, depending on what question you're asking. They are very pleasant to look at, though, and a lot of papers will ask you to make them. It's just very difficult to make interpretations from UMAPs that you wouldn't otherwise get from other clustering-based approaches. That's all. I'm going to leave it politically at that one.
As a
Yeah,
single
and like,
cell analyst, I like no matter how I feel, I'm ordered to make many UMAPs. So I kind of
Everyone
agree,
wants
but
the
just
UMAP. Yeah, we'll
the
get away from
UMAP,
the
just
one
no
day
patience.
maybe, or maybe not. Who knows? Who knows? Maybe it'll just be a pretty thing for the humans to look at. I don't know.
yeah,
We'll
I
see.
guess in any kind of dimensionality reduction, right? I mean, if you're trying to represent 100-dimensional data in compressed 2-dimensional space, then it will be very, very, very hard to do it in such a way that those 2 dimensions are also interpretable.
It's a hard problem. Yeah, I totally get it. I don't have an easy answer for that one. I have gone down some rabbit holes where I've kind of tried to catch up with where they're at with that, and it's yeah, there clearly is a need for a new visualization approach for especially like single cell data, like you mentioned, Emma. And I think if someone can come up with one, they can like really rally people in the field around secondary analysis for single cell in a similar way that like protein ribbon structures. There's some debate around who made it. I'm not even going to start, but they're gorgeous. People love them, and so whenever I talk about proteins, I try to show as many as I can. And they're very intuitive to like look at and understand, even though they don't show quite all of the molecular detail that biochemists might need to kind of go down to the atomistic level, for example. But we need something like that for higher dimensional data and other data types. And I think people underestimate the value of like creative and scientifically accurate and intuitive, visualization in like bringing new people into the field. When I was my very first internship as a scientist was at Coriell Cell Repository in Camden, New Jersey, and I got to use what was called the ANIS Image Stream 2000. Basically, it's like a single cell imaging device. It's way ahead of its time. So you have single cells going through a tube, and they get labeled with these molecules that glow, and you shine certain light at them, basically, and then it would stain different structures inside of them. And so I got to take thousands to tens of thousands of like single cell images back then, and they were gorgeous, absolutely gorgeous. And I stumbled upon this really cool discovery in like summer after ninth grade. But it was actually just the images, not even the discovery, that inspired me and really captured my imagination. And I actually point to that machine and those images as to like why I started in biology. So yeah, and I think I'm not alone. I've heard a lot of other stories like that. So yeah, definitely something to chew on there.
Yeah, science communication is, yeah, go ahead, Emma.
I was gonna say it's really cool. I feel like biology can be more like visual and like can draw people in because it's less abstract in some way. Although, as you say, with like the physics element, like at the base, it is abstract,
Yeah,
But yeah, pretty pictures, many pretty pictures in biology. As many as you
it's so
can.
true. It's so true.
Cool.
Yeah, so true. That does make my job a little bit easier. Yeah, because molecules can be beautiful if you let them be.
I'm gonna stop recording.
Sure.
Yeah,